This infographic models the structural requirements for a neural network. To function properly, it needs precise target ratios: 46% type A, 30% type B, and 24% type C. These are typical tolerances during brain development. The final ratios are tightly controlled and stable. But if we look at the physical mechanism building that network, we see something entirely different. Up close, an individual neuronal growth cone exploring its environment is chaotic. Its movements are highly variable and mathematically stochastic. There is no central director simultaneously managing all of these individual neurons to ensure the final math works out. DNA does not function as a master blueprint projecting a rigid design downwards. To resolve this, we have to recognize the levels problem. The behavior we observe at the level of the macro system is entirely divorced from the behavior of the microcomponents constructing it. If we want to understand how a biological system assembles itself, we have to stop looking for top-down instructions and start looking at the local, energy-consuming agents working at the bottom. We define these components as autonomous agents. An autonomous agent executes simple, local rules based strictly on its immediate environment. It possesses no awareness of the macro system it is helping to build. Consider the mechanics of a professional soccer game. As a match unfolds, 22 individual agents make constant, context-dependent decisions. From the perspective of a fan, the interactions are highly variable, and the final score of any single game is entirely unpredictable. Now look at this chart tracking 10 years of data from the German Premier League. Over a decade, exactly 46% of all games end in a home victory, with a standard deviation of just 2%. The exact same system produces two entirely different realities. To the fan watching a single game, the system is chaotic. To a stadium operator analyzing seasonal aggregates, the system operates with mechanical, predictable precision. Biological development relies on this exact statistical property. Variations in immune cells, the random branching of a neuron, the biased walks of chemical gradients, these all function as massive pools of noisy microevents. In a complex network, micro-level noise is not a flaw that needs to be suppressed. The system actually requires that localized variation to produce statistical robustness at the top. This grid visualizes the Rule 110 cellular automaton. We start with a row of discrete cells. Each acts as an agent, evaluating its neighbors to determine its next state. Hundreds execute this simultaneously. As time progresses, a complex asymmetric macrostructure emerges. No single cell in this automaton possesses the information required to predict the global geometric pattern it is currently constructing. From the internal perspective of the agent, the process is a strict execution of rigid rules. From the external perspective of the observer, the result is deep, chaotic complexity. This proves that a biological process can be driven entirely by deterministic local mechanics, while the resulting structure remains completely unpredictable from the outside. We can observe this swarm logic at scale in the construction of termite mounds. Millions of insects organize to build towering, climate-controlled structures. They achieve this without a master architect through a mechanism called stigmergy, indirect coordination through traces left in the physical environment. Let's map the local rule. An individual termite explores randomly, dropping mud laced with an attractive pheromone. This creates a chemical aura, drawing others. As more agents arrive, their mud compounds the pheromone, creating an exponentially stronger signal that dictates the swarm's movement. The individual termite is entirely oblivious to the team effort. It is merely reacting to the strongest concentration of pheromones directly in front of its mandibles.
Massive structural complexity emerges naturally. You do not need centralized communication if your agents are programmed to alter their environment and react to the alterations of others. The developing brain relies on a biological equivalent of stigmergy. A presynaptic growth cone is blindly extending sensory probes called philopodia into the surrounding tissue. These probes operate under a strictly defined algorithmic rule set known as synaptotropic growth, which governs how dendrites and axons physically locate their synaptic partners. Step 1. Branches explore randomly. Contact with a correct partner stabilizes the connection. Failures retract. Step 2. The stabilized site becomes a branch point for new exploration. Through thousands of iterations, a massive directed structure emerges exactly toward the target partners. Because this branching relies heavily on stochastic, localized exploration, the resulting physical structure of every single neuron is as mathematically unique as a snowflake. This trial-and-error approach is incredibly resilient. It allows the cell to successfully find functional targets in a dark, densely packed, and highly variable physical space. Brain wiring is an active, algorithmic search process driven by localized rules of contact. It is not a static map drawn by genes. If the rules of assembly are so heavily dependent on the local environment, what happens when researchers attempt to isolate these cells in a lab? When you place a single neuron in a sterile petri dish, it will run its baseline cell-autonomous program. If you introduce a chemical attractant, the isolated cell will blindly follow that chemical gradient to its source. This reveals the base mechanism, but it completely ignores the concept of dynamic normality. Dynamic normality describes the natural state of a living brain, where billions of individual algorithms execute simultaneously within an overwhelming density of other active agents. The true algorithm relies on these agents constantly pushing against one another, physically and chemically altering each other's terrain. Extracting a single cell from this matrix deletes the physical constraints and competing signals it requires to make specific decisions. We cannot reverse engineer biological intelligence by isolating a component. The properties that yield precise neural wiring only exist in the stigmergic relationship between the agent and the crowd. So, how does evolution actually program a dynamic, self-organizing jigsaw puzzle of this scale? A genetic mutation alters a single local rule at an early step in the algorithm, but because the entire system is highly context-dependent, we cannot linearly predict what that small change will do. That single altered rule propagates through space and time. It alters the environment for neighboring agents, forcing a cascade of entirely new structural configurations down the line. Evolution does not project the final outcome of this cascade. It simply applies random mutations to the local rules, tests the structural output, and selects the variation that survives. Early cybernetics pioneers, like Ross Ashby, recognized this decades ago. They deduced that to manage this level of complexity, the brain had to function as a highly adaptive, self-regulating machine. Later, AI researcher Marvin Minsky formalized this exact architecture in his seminal framework, The Society of Mind. Minsky argued that true intelligence is not a singular top-down entity. It is an emergent property, built by joining together a massive society of highly specialized microscopic processes that possess no thoughts or awareness of their own.
The absolute robustness of a biological network and the blueprint for resilient artificial intelligence does not require forcing global perfection from the top down. It requires harnessing the localized, chaotic friction of autonomous agents working in the dark.